Science. 14. Cell biology. Intricacies of cellular biology. Unveiling the inner workings of cells. 1. Cell theory. The concept that all living organisms are composed of cells, which are the basic units of life. Cell theory is a fundamental principle in cellular biology. It states that all living organisms are made up of one or more cells, and cells are the basic structural and functional units of life. Cells carry out essential processes such as metabolism, growth, reproduction, and response to stimuli. The development of cell theory in the 19th century by scientists like Matthias Schleiden and Theodor Schwann revolutionized our understanding of life and laid the foundation for modern biology. It provided a unifying framework to study and explain the diverse forms and functions of living organisms, from single-celled bacteria to complex multicellular organisms. 2. Cell structure. Cells have various organelles such as the nucleus, mitochondria, and ribosomes, each performing specific functions. 2. The structure of a cell is highly organized and specialized to carry out its specific functions. Cells contain membrane-bound organelles that compartmentalize different cellular processes. The nucleus houses the cell's genetic material, DNA, and controls gene expression. Mitochondria are the powerhouses of the cell, responsible for producing ATP through cellular respiration. Ribosomes are involved in protein synthesis, while other organelles such as the endoplasmic reticulum, Golgi apparatus, and lysosomes have roles in protein modification, sorting, and degradation. The cytoskeleton provides structural support and enables cell movement, while the cell membrane regulates the exchange of substances with the external environment. 3. Prokaryotic and eukaryotic cells. The distinction between these two major types of cells, with eukaryotic cells being more complex and containing a nucleus. Prokaryotic and eukaryotic cells represent two major categories of cellular organization. Prokaryotic cells, such as bacteria, are simpler and lack a distinct nucleus. Their genetic material is located in the cytoplasm, typically in the form of a circular DNA molecule. Eukaryotic cells, found in plants, animals, fungi, and protists, are more complex. They have a distinct nucleus that houses the genetic material, organized into multiple linear chromosomes. Eukaryotic cells also contain membrane-bound organelles, allowing for compartmentalization of different cellular functions. The distinction between prokaryotic and eukaryotic cells highlights the diversity of cellular life and provides insights into the evolutionary history and complexity of organisms. 4. Cell membrane. This structure controls the movement of substances into and out of the cell, maintaining a stable internal environment. The cell membrane, also known as the plasma membrane, is a crucial component of all cells. It separates the internal contents of the cell from the external environment. The cell membrane consists of a phospholipid bilayer with embedded proteins. It acts as a selectively permeable barrier, controlling the passage of substances into and out of the cell. Small molecules, such as oxygen and carbon dioxide, can pass through the membrane freely via passive diffusion. Larger molecules and ions require specialized transport proteins for their movement. The cell membrane also plays a role in cell signaling, adhesion, and communication with neighboring cells. It helps maintain a stable internal environment, known as homeostasis, by regulating the exchange of materials and nutrients with the surroundings. 5. Cell cycle. The process of cell growth, DNA replication, and cell division that leads to the production of new cells. The cell cycle is a highly regulated series of events that ensures the accurate duplication and division of cells. It consists of several distinct phases, including interphase, G1, S, and G2 phases, and mitosis, including prophase, metaphase, anaphase, and telophase. During interphase, the cell grows, carries out its normal functions, and replicates its DNA. The DNA replication occurs during the S phase, where each chromosome is duplicated to prepare for cell division. Following interphase, the cell enters mitosis, where the duplicated chromosomes are segregated into two daughter cells. The cell cycle is tightly controlled by a complex network of regulatory proteins and checkpoints to ensure the integrity and accurate transmission of genetic material. Proper regulation of the cell cycle is essential for growth, development, tissue repair, and the maintenance of a healthy organism. 6. DNA, genes, and chromosomes. Understanding how genetic information is stored in the cell and passed on during cell division. DNA, deoxyribonucleic acid, is the genetic material that carries the instructions for the development, functioning, and reproduction of all living organisms. Genes are segments of DNA that contain the information for building specific proteins or RNA molecules. In eukaryotic cells, DNA is organized into structures called chromosomes, which are located in the nucleus. During cell division, the chromosomes condense and are segregated into daughter cells to ensure that each cell receives the correct genetic information. 1. The study of DNA, genes, and chromosomes is central to understanding inheritance, genetic disorders, and the mechanisms of microevolution. 2. Protein synthesis. The process of transcription and translation, which turns the information in DNA into functional proteins. 7. Protein synthesis is a fundamental cellular process that enables cells to produce the proteins necessary for their structure and function. It involves two main steps: transcription and translation. Transcription occurs in the nucleus, where the DNA sequence of a gene is transcribed into a messenger RNA, mRNA, molecule. The mRNA then travels to the cytoplasm, where translation takes place. During translation, the mRNA sequence is read by ribosomes, and amino acids are assembled into a polypeptide chain according to the genetic code. The resulting protein is folded into its functional conformation and can carry out specific cellular functions. Protein synthesis is tightly regulated and plays a critical role in determining cell identity, responding to environmental stimuli, and maintaining overall cellular homeostasis. 8. Mitochondria and energy production. The role of these organelles in producing ATP, the main energy currency of the cell. Mitochondria are double-membraned organelles found in eukaryotic cells. They are often referred to as the powerhouses of the cell because their primary function is to generate energy in the form of adenosine triphosphate, ATP, through cellular respiration. Mitochondria have their own DNA and protein synthesizing machinery. They have an inner membrane with numerous folds called cristae, which increase the surface area for ATP production. Within the mitochondria, a series of metabolic reactions, including the citric acid cycle and the electron transport chain, extract energy from nutrients, such as glucose and fatty acids, to produce ATP. This process provides the cell with the energy needed for various cellular activities, including growth, movement, and maintaining ion gradients. 9. Cell signaling. The complex system of communication that governs basic cellular activities and coordinates cell actions. Cell signaling is the process by which cells communicate with each other to coordinate their activities and respond to external and internal cues. It involves the transmission of molecular signals, such as hormones, neurotransmitters, or growth factors, from one cell to another. Signaling can occur through direct cell-to-cell contact or by the release and reception of chemical signals. The signaling molecules bind to specific receptors on the surface or within the target cell, triggering a series of intracellular signaling events that lead to specific cellular responses. Cell signaling plays a critical role in development, immune response, tissue repair, and maintaining homeostasis. Dysregulation of cell signaling pathways can contribute to various diseases, including cancer, neurological disorders, and autoimmune conditions. 10. Endoplasmic reticulum and Golgi apparatus. These organelles play key roles in protein and lipid synthesis and modification. The endoplasmic reticulum, and Golgi apparatus are interconnected organelles involved in the synthesis, modification, and transport of proteins and lipids in eukaryotic cells. The consists of a network of membrane-bound tubules and cisternae, and it has two distinct regions: the rough studded with ribosomes, and the smooth which lacks ribosomes. The rough is involved in protein synthesis, folding, and initial quality control, while the smooth is responsible for lipid synthesis, detoxification, and calcium storage. Proteins synthesized in the are then modified and packaged in vesicles and transported to the Golgi apparatus. The Golgi apparatus further processes and sorts the proteins, directing them to their final destinations within the cell or for secretion. The and Golgi apparatus are essential for maintaining the integrity and function of the cell's endomembrane system and play critical roles in cellular communication, secretion, and the synthesis of membrane components. 11. Cell differentiation. The process by which a less specialized cell becomes a more specialized cell type, crucial for development and tissue repair. Cell differentiation is a complex process that occurs during the development of multicellular organisms and plays a key role in tissue formation, maintenance, and repair. It involves the specialization of cells into different cell types with distinct structures and functions. During differentiation, cells undergo changes in gene expression, leading to the development of specific cellular characteristics and behaviors. These changes are controlled by intrinsic factors, such as transcription factors and epigenetic modifications, as well as extrinsic signals from the surrounding microenvironment. The process of cell differentiation is highly regulated and tightly coordinated to ensure the proper formation and function of different tissues and organs. Understanding the mechanisms of cell differentiation is crucial for regenerative medicine, tissue engineering, and the treatment of developmental disorders and degenerative diseases. 12. Autophagy and apoptosis. Understanding the mechanisms of cellular self-eating for recycling and programmed cell death for removing damaged cells. Autophagy and apoptosis are essential processes that maintain cellular homeostasis and play critical roles in development, aging, and disease. Autophagy is a cellular recycling mechanism in which damaged or unnecessary components, such as organelles or protein aggregates, are sequestered into specialized vesicles called autophagosomes and then delivered to lysosomes for degradation and recycling. This process helps cells remove damaged components and provide a source of nutrients during periods of stress or nutrient deprivation. Apoptosis, or programmed cell death, is a tightly regulated process that eliminates unwanted or damaged cells. It plays crucial roles in embryonic development, tissue remodeling, and immune system function. Dysregulation of autophagy and apoptosis can contribute to various diseases, including neurodegenerative disorders, cancer, and autoimmune conditions. 13. Cell motility. Studying how cells move, through structures like cilia, flagella, and the cytoskeleton, is key to understanding processes like wound healing and immune response. Cell motility refers to the ability of cells to move and change their position in response to internal or external cues. It is a fundamental process involved in various biological phenomena, including embryonic development, wound healing, immune response, and cancer metastasis. Cell motility is driven by complex cellular structures, such as cilia, flagella, and the cytoskeleton. Cilia and flagella are specialized appendages that extend from the cell surface and enable cell movement. The cytoskeleton, composed of microfilaments, microtubules, and intermediate filaments, provides structural support and allows cells to change shape, extend protrusions, and generate contractile forces. Understanding the mechanisms and regulation of cell motility is essential for elucidating the underlying processes of tissue development, repair, and disease progression. 14. Cellular aging. The process of cellular senescence and its implications for age-related diseases and aging. Cellular aging, also known as cellular senescence, refers to the gradual decline in cellular function and viability that occurs with aging. Over time, cells undergo a variety of changes associated with aging, including telomere shortening, DNA damage accumulation, mitochondrial dysfunction, and altered gene expression. Cellular senescence can lead to a loss of tissue homeostasis and function, contributing to age-related diseases such as cancer, neurodegenerative disorders, and cardiovascular diseases. Understanding the mechanisms underlying cellular aging is a critical area of research aimed at identifying interventions to delay or prevent age-related diseases and promote healthy aging. 15. Stem cells. The biology of these unique cells, which can self-renew and differentiate into various cell types, has huge potential for regenerative medicine. Stem cells are indifferentiated cells that have the remarkable ability to self-renew and differentiate into specialized cell types. They exist in various tissues and play a critical role in development, tissue repair, and regeneration. Stem cells can be classified into two main types: embryonic stem cells, derived from embryos, and adult stem cells, found in adult tissues. Embryonic stem cells have the potential to differentiate into any cell type in the body, while adult stem cells are more limited in their differentiation potential. Stem cell research holds tremendous promise for regenerative medicine and treating diseases by replacing damaged or lost cells. Understanding the biology of stem cells, including their self-renewal, differentiation, and regulation, is crucial for harnessing their therapeutic potential and addressing challenges associated with their use. 16. Immunology and white blood cells. The workings of the immune system at a cellular level, including how white blood cells fight off infections. Immunology is the study of the immune system, which defends the body against infections, pathogens, and abnormal cells. At the cellular level, the immune system relies on various types of white blood cells, including lymphocytes, B cells and T cells, phagocytes, macrophages and neutrophils, and natural killer cells. These cells work together to identify and eliminate foreign invaders by recognizing specific antigens, activating immune responses, and coordinating immune defenses. Lymphocytes produce antibodies, mediate cellular immune responses, and maintain immunological memory. Phagocytes engulf and destroy pathogens, while natural killer cells target virus-infected and cancerous cells. Understanding the cellular mechanisms underlying immune responses and the interactions between different types of immune cells is critical for developing vaccines, immunotherapies, and treatments for immune-related disorders. 17. Cellular respiration. The process by which cells extract energy from nutrients, involving glycolysis, the Krebs cycle, and the electron transport chain. Cellular respiration is the metabolic process by which cells convert energy from nutrients, such as glucose, into ATP, the cell's primary energy source. It involves a series of enzymatic reactions that take place in three main stages: glycolysis, the Krebs cycle, also known as the citric acid cycle or TCA cycle, and the electron transport chain. During glycolysis, glucose is broken down into pyruvate, producing a small amount of ATP and NADH. In the presence of oxygen, pyruvate enters the mitochondria and undergoes further oxidation in the Krebs cycle, generating additional ATP, NADH, and FADH2. The electron carriers NADH and FADH2 then donate electrons to the electron transport chain, located in the mitochondrial inner membrane. This process drives the production of ATP through oxidative phosphorylation. Cellular respiration is essential for providing cells with the energy required for various cellular processes and maintaining overall cellular function. 18. Viruses and cells. Understanding how viruses hijack cellular machinery to reproduce, leading to illness. Viruses are infectious agents that can only replicate inside host cells. They consist of genetic material, either DNA or RNA, enclosed in a protein coat. To reproduce, viruses must infect host cells and take over their cellular machinery. They enter the cell and release their genetic material, which hijacks the cellular machinery to produce viral components. The viral components are assembled into new virus particles, which can then infect other cells. The interaction between viruses and host cells is complex and can have significant consequences for human health. Viral infections can lead to a wide range of diseases, from the common cold to severe illnesses such as COVID-19. Understanding the interactions between viruses and cells at the molecular and cellular levels is crucial for developing antiviral treatments, vaccines, and strategies to control viral outbreaks. 19. Transport across cell membranes. The methods cells use to transport molecules across their membrane, including passive transport, active transport, and endocytosis. Cell membranes play a vital role in regulating the movement of molecules into and out of cells. Transport across cell membranes can occur through various mechanisms. Passive transport refers to the movement of molecules across the membrane driven by concentration gradients, without the input of cellular energy. This includes simple diffusion, facilitated diffusion, assisted by transport proteins, and osmosis, the movement of water across a selectively permeable membrane. Active transport, on the other hand, requires the input of energy, usually ATP, to transport molecules against their concentration gradients. Active transport is carried out by specific proteins called transporters or pumps. Endocytosis is a process by which cells engulf molecules or particles by forming vesicles from the cell membrane. It includes phagocytosis, engulfing solid particles, and pinocytosis, engulfing liquid or dissolved substances. Understanding the various mechanisms of transport across cell membranes is crucial for maintaining cellular homeostasis, nutrient uptake, and waste removal. 20. Cancer and cell cycle control. Exploring how failures in normal cell cycle control can lead to uncontrolled cell growth and cancer. Cancer is a complex group of diseases characterized by uncontrolled cell growth and the ability of cancer cells to invade surrounding tissues and spread to other parts of the body. Cell cycle control refers to the regulatory mechanisms that ensure proper progression through the cell cycle, including cell growth, DNA replication, and cell division. Failures in cell cycle control can result in the accumulation of genetic mutations and the uncontrolled proliferation of cells, leading to the development of cancer. Understanding the molecular mechanisms underlying cell cycle control and how they are disrupted in cancer cells is crucial for the development of targeted cancer therapies and strategies for cancer prevention and early detection. 21. Cell junctions and extracellular matrix. How cells interact with each other and their environment is crucial for tissue structure and function. Cell junctions and the extracellular matrix, ECM, play essential roles in cell-cell and cell matrix interactions, providing structural support, coordinating cell behavior, and contributing to tissue organization and function. Cell junctions are specialized structures that connect adjacent cells and can be classified into three main types. Tight junctions, form a barrier to prevent substances from passing between cells, adherens junctions, mediate cell-cell adhesion, and gap junctions, allow direct communication between cells. The ECM is a complex network of proteins and carbohydrates that surrounds cells and provides mechanical support, regulates cell behavior, and helps maintain tissue integrity. It consists of proteins such as collagen, elastin, and fibronectin, as well as proteoglycans and glycosaminoglycans. Understanding cell junctions in the ECM is crucial for unraveling the mechanisms of tissue development, wound healing, and diseases such as cancer and fibrosis. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts.